In the last class we completed the almost all part of the genetics. In this class we have to discuss a small part of Human Genome Project and very important part, that is the evolution. Regarding the EAMCET point of view, from the Human Genome Project we may or may not get one question, but from the evolution part we are getting 3 marks questions. That means we are getting the 3 marks from the evolution. Regarding the evolution, from the Hardy-Weinberg equilibrium we are getting one question. Mechanism of evolution to the end of topic we are getting one question, and from the introductory part to 2 up to the theories we are getting one question. Again repeating, regarding the evolution, in the EAMCET examination we are getting 3 questions. One question from the introduction up to theories, Lamarckism, Darwinism, mutation theory, and one question from the Hardy-Weinberg equilibrium, and the third question, mechanism of evolution to the end of topic. What about the Human Genome Project? From the Human Genome Project we may or may not get one question. As we observe the Human Genome Project, in our body 23 pairs of chromosomes are present, or 46 chromosomes. In the 23 pairs of chromosomes, 23 pairs of DNA strands are present. Now as we observe the genome, what is meant by genome? Only 23 chromosomes, that means haploid. We have to discuss Human Genome Project with reference to only 23 chromosomes. As we observe these haploid set of chromosomes or 23 chromosomes, near about 3.5 million nucleotide base pairs are present. Again repeating, as we observe the genome, that is a haploid set, that means 23 chromosomes. On 23 chromosomes, near about 3.5 billion nucleotide base pairs are present. Already the well established persons conducted experiments on Caenorhabditis, Arabidopsis and E. coli. Again repeating these 3 words, the scientists, the eminent personalities already completed the genome project regarding the E. coli bacterium, Caenorhabditis, that is a nematode, E. coli, that is a bacterium, and Arabidopsis, a plant. On these 3 organisms, the eminent personalities successfully completed the genome project. Based on that confidence, they started Human Genome Project. Regarding the Human Genome Project, almost all the personalities successfully completed their work. Now as we observe the Human Genome Project, in the Human Genome Project we have to discuss the gene mapping. In the gene mapping we have to study the C band, the G band, low resolution, high resolution, macro restriction and contig methods. Again repeating, as we observe the mapping, one is gene mapping and the second one we call it as physical mapping. Then we have to discuss the low resolution. In the low resolution, the C band, G band, fluorescent methods. In the high resolution, we have to study the macro restriction and contig methods. Generally, as we observe the low resolution, whenever the base pairs are more in number, we go for the low resolution. Whenever the base pairs are more in number, that means if the base pairs are more in number, we go low resolution. If the base pairs, if the number varies, we go for the high resolution. So whenever the base pairs number is less, we go for the high resolution. If the base pairs are more in number, we go for the low resolution methods. In the Human Genome Project, one method we call it as the DNA fingerprint technology. And after the DNA fingerprint technology, what is the gene therapy? And these points in briefly we have to discuss. Now we go for the first question. Now as we observe the first question, DNA fingerprint technique was first developed by. Now as we observe the DNA fingerprint technology, as we observe the human beings or the living organisms, in all organisms the arrangement of base pairs is not one and the same. Person to person, the arrangement of base pairs varies. That we call it as such difference in the arrangement of base pairs is called the DNA fingerprint technology. Now here the question is, DNA fingerprint technique was first developed by Jeffries et al. That means associated with Jeffries and others also worked on this technology. DNA fingerprint technology means in all living organisms, for example human beings, one person to another person, as we observe the arrangement of base pairs, in the number of base pairs varies. Such variation in the base pairs number is the DNA fingerprint technique that was first proposed by Jeffries. So the answer is, go for the answer is Jeffries. He worked on the gene responsible for the protein which is for the muscle, that we call it as the myoglobin. Now we go for the next question. As we observe the next question, polymerase chain reaction is used for. What is meant by polymerase chain reaction? That is PCR. We call it as polymerase chain reaction. That is used for, one option is DNA synthesis, the second one is DNA amplification, third one is protein synthesis, and the next one is amino acid synthesis. And we know very well, as we observe the DNA synthesis, for example this is double stranded DNA. During the formation of new strands, both the strands are separated. For the DNA synthesis, each strand works as a template. So each old strand synthesizes a new strand. That concept is separate, that we call it as DNA synthesis. And protein synthesis we know very well. Now as we observe the DNA strands, one is 5' 3', one is 3' and 5'. During the protein synthesis, the 3' 5' strand, that we call it as template. From this template, by a process called transcription, we are getting mRNA. The mRNA by a process called translation gives rise to polypeptide chain. A group of amino acids that we call it as polypeptide chain. So automatically the polypeptide chain becomes one protein. The proteins are chemically made by amino acids, that is a separate concept. Now here PCR, polymer chain reaction, is used for what purpose? DNA amplification. As we observe the DNA amplification, two strands are present. As the temperature increases, the two strands are separated. then we use the probes. The probes are going to attach these two templates where the correct matching is present. Until that, the probes between the two templates, they perform a movement that we call it as the Brownian movement. It means in the DNA amplification, we discuss three stages. One is denaturation, and the second one is annealing, and the third one is extension. So here polymerase chain reaction is used for, the answer is DNA amplification. Now we go for the next question. As we observe the next question, in DNA fingerprinting, the analysis is made for, as we observe the DNA fingerprinting, the analysis is made for example, this is one DNA strand. In this DNA strand, here you observe 1, 2, 3, 4, 5. Here how many A's are present? 5. Now it is followed by the G. Here how many are present? 2. And here as we observe 1, 2, 3, 4, 5, 6. That means here they are the C. 1, 2, 3, 4, 5, 6. 1, 2, 3, 4, 5, 6. And it is followed by A, G, C and T. Now here 1, 2, 3, 4, 5. 1, 2, 3, 4, 5. Now the same strand is repeated. Now in this place, in the first place, this indicates first place. Here how many A's are present? 1, 2, 3, 4 and 5. Now as we move, here how many are present? 2. Here A. How many are present? 3. Here 2. Here 5. That means in a DNA strand, as we observe the arrangement of base pairs, there is variation in the arrangement of number of base pairs. That we call it as variable tandem number repetitives. Variable tandem number repetitives. We also use VTNR. Here in a strand, in this place, how many times here repeated? 5. Now as we move forward, here how many times repeated? 2. Here how many times repeated? 3. Like that in a strand, the base pairs arrangement varies from place to place, from person to person. Now here as we observe in DNA fingerprinting, the analysis is made for variable number tandem repetitives. Variable tandem number repetitives. Now we go for the next question. Matching DNA sequences of criminal with suspect is known as, generally the DNA fingerprint technology is used for the conservation of wildlife and in order to solve medico-legal cases, and it is used in the study of anthropological studies, and it is used in the classification of animals, etc. And it is also used in the pedigree analysis. What is meant by pedigree analysis? To know the ancestral history of an organism in order to solve. For example, as we are going to meet one murder or we are going to meet one rape case, in order to solve such cases, generally the investigated team personalities follow what type of technique? That we call it as DNA fingerprint technology. Because from person to person as we observe the DNA strand. In the DNA strand, the arrangement and number of base pairs is not the same, varies from person to person. That we call it as variable tandem number repetitives that plays an important role in the DNA fingerprint technology. So that here, what is the answer is the DNA fingerprinting. Now we go for the next question. Retrovirus has the genetic material. In order to carry the gene, we have to use different vectors. Generally, we know very well what is the most popular vector, that is PBR322. In the biotechnology part, we learned what are the vectors. One we call it as plasmids, one we call it as bacteriophages, one we call it as cosmid. With the help of plasmid, we can introduce only small part of DNA strand. With the help of bacteriophage also, we can introduce only small strand of DNA. But by mixing the plasmid and bacteriophage, we are getting the cosmid. With the help of the cosmid, we can incorporate the entire length of DNA strand. In addition to the cosmid, we have to use back and we have to use yak. In the same way, what type of vectors are used to incorporate the gene or the DNA? That we call it as the vectors. One we call it as retrovirus. In the retrovirus, we know very well, one example for the retrovirus is HIV virus. In the retrovirus, what type of genetic material is present? Single stranded RNA is present. So the answer is the ssRNA means single stranded RNA. Now the next one is, as we observe the next question, adenovirus has what type of genetic material? Adenovirus, it has DS, double stranded DNA is the genetic material in the adenovirus. Now we go for the next question. Adeno-associated virus. In the first we discussed retrovirus, single stranded RNA. In the last question, adenovirus, double stranded DNA. Here adeno-associated virus has what type of genetic material? Single stranded DNA. If it is ssRNA, retro. If it is dsDNA, that is adenovirus. If it is ssDNA, that is adeno-associated virus. Now the answer is single stranded DNA. Now we go for the next question. DNA is protected from the damage by. DNA is protected from the damage by. Generally in the gene therapy, we have to introduce the gene. Now this is the gene. Gene means it is the part of DNA. In order to introduce the DNA into the host cell, to protect this gene, it is coated by lipoplex or it is coated by polymers. Because of the presence of lipid materials or because of the presence of polymers, the gene is being protected during the gene therapy. Now here DNA is protected from the damage by. The answer is, we go for the lipoplex. The answer is lipoplex or it is also coated by the polymers. Now the next one. Genes involved with the disease or inactivated by. As we observe the Human Genome Project, majority of the diseases are genetical based diseases. One example is the hemophilia. One example is the color blindness. All these are the genetically based diseases. We know very well as we observe the hemophilia. In the hemophilia, we learned hemophilia A, hemophilia B, hemophilia C. In the hemophilia A, what type of factor is deficit? That is 8th factor. In the hemophilia B, 9th factor. In the hemophilia C, the 11th factor. It means these factors produced gene is damaged. How such diseased gene is rectified? Here the question is, genes involved with the disease are inactivated by. The answer is synthetic oligonucleotides. We know very well phages. That is nothing but bacteriophages, one type of vector. Vectors, there are many types. One is phasmids, one is bacteriophages, one is cosmid, and the recent vectors are bat, yak, etc. and lipoplex. That means the gene is being coated by lipids or the gene is being coated with polymers. Here the gene associated with the disease can be treated or rectified by introducing. The answer is the synthetic oligonucleotides. Now the next question is, Adenosine D Amines enzyme, ADA. Now Adenosine D Amines enzyme is associated with what type of disease? Secondary combined immunodeficiency. The one who has been suffering from this disease, in his body the enzyme is deficit. That means the one who has been suffering from this disease, in his body the gene unable to produce ADA. That means ADA producing gene got disease. Based on this disease, the scientist introduced the gene therapy. The gene therapy is of the two types. One is somatic gene therapy and germplasm gene therapy. But we are not practicing the germplasm or the germline gene therapy, because in the germline gene therapy the gene incorporated with the allosomes or sex chromosome. So in the coming individuals we are getting some problems. So the germline gene therapy is being stopped. We are going for the somatic gene therapy, and it is in vitro in vivo. That means the gene removed from the body and rectified, later inserted into the body, and in some cases the damaged gene rectified inside the cell, inside the body. Now here adenosine D amines enzyme is associated with what type of disease? That we call it as SCID, secondary combined immunodeficiency. Now we go for the next question. For blotting, one of the following is used. As we observe the DNA fingerprint technology protocol, now this one we call it as a beaker. Now in this beaker, this one we call it as the cork made bed. This is cork made bed. Now on this bed we have to arrange a nylon membrane. This is nylon membrane. In order to escaping the nylon membrane, above nylon membrane we kept the towels, that means blotting papers. Now in this beaker we take agar sal gel. Now we introduce the DNA strand. Now this is the DNA strand. Now we apply restriction endonuclease enzyme. Now the restriction endonuclease enzyme, it cleaves, it cut the DNA strand at palindrome sequence. Now in the beaker, in the agar-zole, what are present? Small fragments and larger fragments are present. Now this is one fragment, the two strands are separated. Now we use the radioactive properties probes. Now what is going to happen? These probes, they are single stranded. Now as we observe the probes, these probes having radioactive properties. Now we introduce the probes into the beaker. Now these probes linked with the strands having the complementary information. Now what is going to happen? In the beaker, above nylon membrane, what are present? The blotting papers are present. Now the blotting papers slowly absorbs the agar-zole. While the blotting papers slowly absorb the agar-zole, these fragments slowly shift on nylon membrane. Now this is the nylon membrane. On nylon membrane, as we observe, the fragments are present. In order to get the clarity, we apply X-ray film. Now the chemical reactions taken place. Now as we observe the X-ray film, on X-ray film, thin strand, thick strand, thin strand, thick strand, like that we have to observe the prints. That is called the DNA fingerprint technology. If the thin strand present, there the number of base pairs less. In the thick strand, the number of base pairs are more. Now here, for blotting, one of the following is used: plasma membrane with paper towels, nylon membrane with paper towels, cellulose membrane with paper towels, agar-zole membrane with paper towels. Here agar-zole, that is a gel, it is not a membrane. Now here what we used? Nylon membrane plus blotting papers or towels. Now the answer is the nylon membrane with paper towels. Now we go for the next question. Now abiogenesis. What is meant by abiogenesis? According to the abiogenesis, the living organisms derived from non-living organisms. This abiogenesis theory supported by great personalities such as Aristotle, Plato, Thales, Van Helmont, etc. supported abiogenesis theory. Now here, abiogenesis theory was believed by Aristotle, Plato, Thales and Van Helmont. Now the answer is the Van Helmont. Now the next question. Who among the following disproved abiogenesis theory? What is meant by abiogenesis? living organisms derived from the non-living organisms. No, it is not the correct. It is discarded or disproved by Francesco Redi, Spallanzani and Louis Pasteur. Experimentally it is disproved by the Louis Pasteur. But here, who among the following disproved abiogenesis theory? Aristotle, he supported abiogenesis. Van Helmont supported abiogenesis. Thales supported abiogenesis. And Spallanzani. Now the answer is the third one, Spallanzani. Now the next one. As we observe the next one, assertion. In Swan neck flask experiment, meat broth remained free of microbial contamination for many months. Now, as we observe the Swan flask neck, now the Swan neck flask experiment, this experiment conducted by Louis Pasteur. Now this is Swan flask experiment. Now here the neck is narrow and this is the mouth opening, and they have taken the meat broth. Now the opening is not covered by any covering. The microorganisms entered, and these microorganisms are trapped in the narrow neck. So the meat broth which is present in this apparatus not underwent any decomposition, because the microorganisms are trapped in the narrow neck. Here the opening is left. The opening is left unsealed. That means not covered. Now here you observe, in Swan neck flask experiment, meat broth remained free of microbial contamination for many days, because the microorganisms are trapped in the narrow neck. Now here the reason, flask used in this experiment was left sealed. No, it is not the correct. The opening is not covered by any proper covering, so it is left unsealed. That is correct. Here what they gave, flask used in this experiment was left sealed. So A is correct, R is wrong. Now we go for the next question. Arrange the following phases of origin of Earth in a sequence. Now in the primary classes we are having one idea that we call it as the solar system. In the solar system, the head we call it as the sun. Now this one we call it as the sun. Around the sun the orbits are present. Now this one we call it as orbit. Now in the orbit the planets are moving. Now as we observe the planets, 9 planets are present. Name the planet on which the life is present, the Earth. Now here what our question is, how the planets are developed. Now here arrange the following stages of origin of Earth in a sequence. Now as we observe, the first one is solidification of planets. Second, contraction and flattening of solar nebula. Third one, fragmentation of interstellar cloud. Now this one we call it as the cloud, interstellar cloud. Now it has undergone the fragmentation. That means this interstellar cloud divided into smaller fragments. Now these fragments undergo contraction and flattening. After contraction and flattening, we got the meteoroids and proplanetary materials. Now finally we got the planets. Now the interstellar cloud underwent fragmentation. It is divided into number of fragments. These fragments undergo contraction and flattening, so that we are getting the meteoroids and proplanetary materials. Finally we got the solidification of planets. Now you observe, here the first one is fragmentation of interstellar cloud, and the next one contraction and flattening of nebula. Third one, condensation of nebular material into meteoroids and proplanetary bodies. Finally we are getting the solidification of different planets. So the answer is the third one. Now we go for the next question. Primitive atmosphere was reducing atmosphere. Now as we observe the formation of atmosphere, the atmosphere has 4 layers. One is troposphere, stratosphere, ionosphere, exosphere. But in the beginning of evolution, such 4 layers were absent. In the beginning, in the atmosphere, no oxygen is present. In the beginning, the atmosphere is said to be reducing, because in the beginning in the atmosphere, what is present? The hydrogen is more. Here primitive atmosphere was a reducing, because hydrogen was abundant of all the gases in the atmosphere. So here the answer is both are correct and R explains. Now students, you have to listen. Right now in the universe, as we observe the atmosphere, what is present? The oxygen is present. Now you have to imagine, in the beginning of evolution, if oxygen was present, what happened? There is no question of the formation of life. For the formation of life, raw materials were present in the beginning of evolution. Suppose in the beginning of evolution, if oxygen was present, what was happened? Because of the presence of the oxygen, all raw materials were burned. So there is no question of the life. So in the beginning of the evolution, in the atmosphere, the hydrogen is more, so that the raw materials underwent so many reactions. Later what obtained, that is the life. Now the next question is, choose the correct statements regarding the protobions. What are the protobions? First formed bions. In the protobions, the metabolic activities are slow. What are the protobions? That means conservatives. One is conservatives and microspheres. These two are said to be the protobions. Now here, choose the correct statement regarding the protobions. First statement, all protobions were membrane bounded. No. What are the protobions? One is microspheres, one is conservatives. As we observe the microspheres, they are membrane bounded protobions. In the beginning, conservatives not having any membrane. Later they developed the membrane. So here all protobions were membrane bounded molecules. No, it is wrong. Now, the next one, microspheres, they underwent reproduction or they multiply by what type of process? Budding. Budding come under the asexual reproduction. Now, what about the conservatives? They are also one type of protobions. Conservatives were suspended colloidal systems in the hot dilute soup. Who called the hot sea water as hot dilute soup? Haldane. Now here the answer is. Now the next one is, choose the correct statements regarding the earliest living organisms. What are the earliest living organisms? The earliest living organisms were believed to be the bacteria. So here, the earliest living organisms similar to that of bacteria, so that they come under what type of kingdom? That we call it as Monera. Now as we observe the earliest living organisms, in the earliest living organisms, 1 or 2 DNA molecules are present. They resemble the genetic material of the bacteria. Now the third statement, they obtained energy by the fermentation of organic molecules, because in the formation of earliest living organisms in the atmosphere there is no oxygen and there was no photosynthesis. How the earliest living organisms got energy? By a process called fermentation of organic molecules, and the earliest organisms might be bacteria, so come under the Monera. Like that of the bacteria, the earliest living organisms having 1 or 2 DNA molecules resembles the genetic material of bacteria. So based on this information, what is the answer? Now all given statements are correct. Now the next one is, the early organisms which used inorganic molecules for their energy and carbon dioxide as a source of carbon, because as we observe the evolution of earliest organisms, in the beginning there is no oxygen. How they prepared their food materials? By a process called chemosynthesis. In the chemosynthesis, they use energy derived from the chemicals. So here, the early organisms which used inorganic molecules for their energy and carbon dioxide as a source of carbon. So such organisms are called anaerobic heterotrophs, aerobic heterotrophs, chemoautotrophs and photoautotrophs. Now you observe the answer is the chemoautotrophs. Now the next one, arrange the following organisms in the sequence of their evolution. Now you observe the options. One is chemoautotrophs, second one is anaerobic heterotrophs, third one is aerobes, and the fourth one is photoautotrophs. During the course of evolution of living organisms, first we learned the protobions. Protobions are microspheres and conservatives. In these two, microspheres are membrane bounded organelles. Later conservatives developed the membrane around their body. In the protobions, the metabolic activities are at slow rate. Now they are followed by the living organisms. What are the first type of living organisms? Anaerobic heterotrophs. In the beginning there was no oxygen and all are heterotrophs. they were unable to prepare their own food material. They depend on others. Now, anaerobic heterotrophs followed by chemoautotrophs. Just we completed chemoautotrophs. They prepare their food materials by using energy derived from the chemicals. Now, later photoautotrophs. As we observe the photoautotrophs, they trap radiant energy, and such radiant energy by photosynthesis is converted into the organic material or food material. During photosynthesis, what is released? Oxygen released, and what is utilized? Carbon dioxide. So that after the development of photoautotrophs, the oxygen started releasing into the atmosphere. Then only in the atmosphere we explained the 4 layers, that is the troposphere, stratosphere, ionosphere and exosphere. After the evolution of prototrophs, oxygen released, so slowly the aerobes evolved. Now the answer is